This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 979, recorded on January 27th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. 59 degrees and overcast. We've had near freezing temperatures the last couple of nights, and it's going to be kind of, well, if I look out, at the bogus 2-week forecast, we got stuff between high 40s and mid 60s and some rain, which would be good. So it's winter, or what passes for winter here in Austin.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 27 Fahrenheit, -3 Celsius, and it's cloudy, and we got a dump of about 7 or so inches of snow day before yesterday. So we have snow, but the streets are cleared, sidewalks are cleared, because it's been close to 30. 30, even above 30 yesterday, some, yeah. So 30.
And from Western Massachusetts, Alan Dove.
Good to be here. It's pretty typical, actually a little warmish January day, 39 Fahrenheit, 4C, and broken overcast rolling in. I think we're supposed to get a little bit of snow, but not anything that's going to stick. Yeah.
A couple of announcements for you before we dive into our science today. The first is the research assistant position in the laboratory of Amy Rosenfeld at Center for Biologics Evaluation and Review. That's at the FDA, where they do some research, and Amy has a lab there. She wants an assistant to help out with work on enteroviruses, genetic, biochemical, cell-based studies, development of animal models, all BSL-2 research, and the information will be on a PDF in the show notes. You can find out more about the position, and Amy's email is there to contact her if you are interested. And Kathy has some information about ASV.
Right. So the abstract deadline for the American Society for Virology meeting in 2023 is Wednesday. So this is going to be released on Sunday, January 29th, and 3 days later is when abstracts are due. If you want to apply for a travel award, you're a trainee or teacher of undergraduate virology, those are due 2 days later on Friday, February 3rd. And for some of these things, you need to have membership in ASV. So even if you're procrastinating or getting that last minute data for your abstract, you could spend some time while your blot is incubating and go apply for your membership now. And then I will tell people that even once you've applied for your membership and you submit your abstract, you can go ahead and do your travel application. There's a delay while the membership committee deals with a flood of memberships applications, and we will still count it as you have applied and everything if you do it before the deadline. So you don't need to write to the ASV office and say, please process my membership now, because you're just going to be in a line of people. Anyway,
they will log it as received, in other words, and they'll
exactly
sort out that you applied in time. Yeah,
exactly. So anyway, yeah, Wednesday, February 1st for the abstracts, and Friday, February 3rd for the travel award applications. Memberships, you can start right now and do that. And it's going to be a great meeting. Information is on the web, and look forward to having you come to Athens, Georgia, June 24th to 28th.
When you said flood, I immediately thought there was a flood in the facility.
No, flood of applications, though, for sure.
Yeah, they're not paper anymore, so it wouldn't matter.
Right.
It was a flood.
Right.
All right. One more thing. For a long time, people have been asking us for a Discord server. Those of you who don't know, it has nothing to do with being unhappy. It's just a place where you can go chat. And we started one for Microbe TV, so you can go there and talk about all the different podcasts that we do. And the link will be in the show notes. It's free. You can go. It's 24/7. There's always somebody there because it's all around the world. And people talk about viruses, bacteria, epidemiology, immunology, neuroscience. So check it out.
And there's an off-topic channel, so if you decide you just have to post a meme of something, you can toss it in there.
There are over 200 people there so far, I think, right? In fact, maybe 250. Let's take a look. See if it doesn't take a long time to load here. It's too long. Makes me put a password in. Alan will find out.
Oh, yeah, I'll get to it.
All right. For our snippet today, I wanted to give you an update on poliovirus in the U.S. Now, last summer, there was a case of paralytic polio in Rockland County, New York. This was in an individual who is part of a community that is highly under-vaccinated for polio. I think it was the community had something like 50% vaccination coverage as opposed to 90+% in the rest of the country. And just to remind you, the U.S. uses inactivated polio vaccine exclusively. And this is an activated preparation that's injected into your arm muscle. And it gives you very nice immunity, but it doesn't protect your gut against infection. And I have a piece of data to prove that in a moment, which is very cool. So the result is that much of the rest of the world, at least where the WHO is doing eradication campaigns, they use oral poliovirus vaccine developed by Albert Sabin, who, by the way, died 30 years ago in March. And that virus reproduces in your intestine, of course, gives you wonderful immunity, but reverts in every person, and those revertants spread. And if your guts are susceptible, you can get infected, but you won't get polio if you're vaccinated against IPV. And so I think for many years there have been viruses in the U.S. wastewaters. And
but if you're unvaccinated, those revertant viruses can cause paralytic polio, and that's actually the chief downside of the oral vaccine.
And in fact, we have a lot of type 2 vaccine-associated polio in Africa because vaccination rates drop, and that virus has been around for a while, and it spreads very well, actually.
It works. I mean, it seems to work fine if you go and you're vaccinating everybody, and since everybody's gotten the drops, they are immune and nobody gets paralyzed except in extremely, extremely rare cases. But yeah, if vaccination rates are scattershot or inconsistent, it can be a problem.
And it turned out that the type 2 vaccine-derived polioviruses that are shed, these are revertants that are shed, they transmit very well and spread very well. So they're all over the world. And wherever, as Alan said, wherever vaccination rates drop, you can have paralysis. So there's been 600 cases of paralytic disease in the past year, mostly type 2 vaccine-associated disease. Now, interestingly, wild type 2 as well as wild type 3 have both been eradicated. So we've replaced wild type 2 with vaccine-derived type 2. And you can't get rid of it as long as you're using OPV, right?
Right.
So in Rockland County, vaccination rates were low. Boom, virus comes in. It's probably had been there for a long time. So in response, the CDC finally started doing some wastewater surveillance. And back last summer, when the case arose, they immediately looked in waters around here and they found poliovirus. In fact, a virus, type 2 vaccine-related poliovirus, related to the patient's isolate. And it was found in other waters around the area as well. So, and we talked about this, and I mentioned that it was likely that there's probably vaccine-derived polioviruses in many locales in the U.S. But for years, the CDC has not looked. And in fact, I found a website at the CDC. If you just look for wastewater and polio, they have a little page here, wastewater testing of polio is a well-established tool for polio surveillance. However, it's not recommended for countries such as the U.S. that have eliminated polio. Why isn't it recommended? Because if they find it, well, this is my guess. Now, if they find it, they won't know what to tell you. So
There
they haven't
are a
looked.
few, yeah. So there are a few things going on here. First of all, it is an additional expense and process that has to be done. It has to be done properly, and you have to go to public works departments and justify what you're doing. CDC has a limited budget. Doesn't make sense for them to necessarily do this everywhere all the time. And at the same time, as Vincent just said, if they do find something, well, now you have to tell the public there's polio in the water, and there probably was all along, but then you've got to manage the whole public relations fallout of that.
Yeah. So now they've decided to expand their wastewater testing. And there's a document we will link to, CDC planning wastewater testing in select communities. All right. And so they're doing it only in certain places, one of which is Michigan, and another is Philadelphia, and a few other states as well, as we'll tell you in a moment.
Well, and the New York area is
The
continuing.
New York
right?
area as well.
Yeah.
And they say particularly in places where vaccine coverage is low, which makes sense because there people are at risk. And if you do find polio in wastewater, then you have to get everybody vaccinated. Although my understanding is that they haven't made much of a dent in the Rockland County vaccination rates. That community doesn't want to be vaccinated, so it's going to be hard to do that. So I found in another document a graphic of the wastewater testing results. And I'll put this in the video version for you to look at because I'm going to describe it. It's going to be, I can summarize it though. But what it is, on the left-hand side, it's entitled Polio Wastewater Test Results by MMWR Week. 2,000 tests. This is January 9th, 2023. And on the left is the location. So we have lots of New York locations, New York City, a bunch of counties in New York State like Rockland, Putnam, Orange, etc. And then at the bottom, we have Connecticut, New Jersey, Oregon, and Utah. And then next to those are the number of samples. So there's 678 in New York City, hundreds in the different counties. In Connecticut, we have 87, New Jersey 32, Oregon 11, and Utah 71, which are kind of paltry, small numbers, right? All right, and so then to the right is a line graph of weeks. So they have 0 to 55, so that's a year. And roughly in the middle of this would be the Rockland County isolate. So it's green. There's a green bar if they've taken a sample. And there are lots of gaps here where they haven't taken samples, right? And then there are bars of different colors. For example, so the green is actually PV negative. So if they've taken samples and there's no poliovirus genomes there. Then there are blue bars, which is called poliovirus indeterminate. They don't have enough sequence to tell what it is. Then they have a red bar, which is polio 2 positive, linked to case. In other words, the genome sequence matches or is close to the case to indicate that they're epidemiologically linked. So there are a bunch of red bars throughout New York City, and all the counties. You can see a lot of red bars. So this virus is in many different, I don't know what you call them, sewage processing plants, I guess.
Sewer sheds.
Sewer sheds.
Which is not a shed in which you put sewage. It's like watershed.
Right.
So they talk a bit about this in their guidelines document, considerations for wastewater testing, where they talk about you want to sample a sewer shed from an area that is a sufficient number of households to maybe have a positive result, but not so many households that your sample is going to be so diluted that you wouldn't detect if it is there. And of course, in some urban areas, especially with old infrastructure like New York and Philadelphia, this is complicated by the way the system has grown. You may not be able to sample the right size sewer shed.
Mm-hmm.
So these are different sampling points in the the treatment system.
So, by the way, the poliovirus indeterminate means they don't know what serotype it is. Not enough
Right.
sequence.
They know it's poliovirus.
I know it's poliovirus,
That's it.
but that's it. All right, then we have PV negative, which is green. Then red is PV positive linked to case, as I said. There are a bunch of those in the New York area, but not in not in Connecticut, New Jersey, Oregon, Utah. Then we have pink, PV2 positive, not linked to case. Aha,
It's
it's a
just
separate
there.
introduction, right? It's genetically distinct. So remember, these OPV2s are coming from other countries because we don't use it here. So, and that's not surprising. People are coming in every second, right?
And so there's one of those in New York City, and then there's one of those in Utah.
Yes. Which are, yeah, who knows the relationship between the two. And of course, Utah is very undersampled, so there could be even more. And the Utah samples seem to be from the beginning of last year, I guess. If that, because the Rockland County case is kind of in the middle between weeks 25 and 30. So I guess those are old samples. All right, so that's pink. And then purple is PV2 positive, indeterminate linkage to case. And again, there's some of the purples throughout New York City in one of the counties.
So as distinct from the other indeterminate, one is poliovirus indeterminate, but so indeterminate that you don't even know what type it is.
Correct.
This is PV2, type 2,
Right.
but nevertheless indeterminate relative to the relationship to the case.
Right. So they
Correct.
don't have enough sequence to say this definitely came from this patient we know about, but it could have.
Right. So all in all, I count 4 separate, and then there's a yellow box here.
Yeah,
I'm not-
what is
I
that?
don't know what that is.
There's nothing in the legend about it.
There's no legend to yellow. Yeah, I don't know, but it's not a negative, so it's
That's
either.
there to make sure we're paying attention.
Right.
So you can count 4 independent introductions really, with not a lot of sampling outside of New York. So there could be more. That's the point I'm trying to make. So, what's the solution? Keep the vaccination rates up. We have
So
to
wait a
stay
minute.
Vaccinated.
You said 4 independent introductions.
All right, let me make sure that's correct. So we have the original one, which is pink, and okay, then the Utah one is also. Wait a
No,
minute.
pink is not linked to a
To the
case.
case.
Yeah, so there's one in New York City, pink, that's not linked to the case.
Right.
There's one in Utah that's not linked to the case. There's a purple one in Orange County, which is indeterminate linkage to the case. So that could be a separate introduction also. And then the yellow one, I'm counting
So
as
we don't know
4.
what that is.
We don't know
Okay.
what it is.
So at least 3. I see what you mean by independent introduction.
Yeah,
then.
yeah.
They could not, they're very unlikely to be related.
Right.
Right.
Okay, so this is ongoing, and presumably they will update this. I'll figure out where this came from. I don't recall, but it is a public document. I'll put a link in the show notes.
However, it's pretty clear to me from looking at this that in the counties proximal to Rockland County, where the paralytic case came up,
Yeah.
there's a lot more activity, and specifically this particular virus, than elsewhere. Is that a fair statement?
It's a lot more sampling there too, right?
Well, but
well,
if you look at
don't see-
Utah
you
has,
don't see any-
look at
yeah.
Sullivan County. That's only 48 samples.
And you don't see any red bars anywhere outside the New York area. So you don't see any link to the case in Connecticut or New Jersey, for example.
Right.
And actually, it's interesting. I just noticed this, that they're clustered in time as well.
That's right.
Because
Yes.
after about 43 weeks, except for one little red blip in Orange County that overlaps the yellow blip,
It's
it
gone.
disappears.
That's
Yeah.
right. That's
That's
right.
Which struck me because back before the vaccines, there was a polio season in the summer,
Yeah.
right? And
Should
I don't
still
know.
be, right. It should still be the case.
It
right?
should still be the case. It's spreading. It's a revertant virus. It was a vaccine for a long time, and now it's not.
Yep.
And it's it quit that business and got back into the old activity. I guess it would spread in the summer for the same
Yeah.
reasons it always spread
Sure.
in the summer, right?
Yeah.
So one of the cautions, I think, in this article and in the CDC links that you put up is that aside from identifying which sewer shed these are in, you can't pin it down geographically any further than that.
Yeah.
Okay? So I would say the most likely thing is that the knee-jerk reaction is that the guy who got the paralytic polio is the index case, but no, he's a bystander probably. All right.
Oh yeah, many people were infected, but he got paralyzed. Yeah.
Right. They're probably that's the important thing. Many people were probably contributing to this.
Right.
There was an outbreak, if you like, of this geographically nearby.
Right.
So where's Putnam? Well, no, I guess there was nothing in Putnam County. I'm looking at this. Rockland, Orange, and Sullivan counties, where most of the stuff are all proximal to each other.
Yeah, that's all just up the Hudson, bedroom communities for New York City.
Nassau County has one hit. That's further out, right?
It's Long
That's
Island.
Long Island.
Long Island.
So that's on the other side of the city.
Right. Okay.
Yeah. So the thing is that this idea of it coming and going, we should have more sampling to know that, right? So what are the patterns? What are the epidemiological patterns? Is it just in the summer, or does it happen all year round? I think that for that reason, it's worthwhile doing more sampling as opposed to what the CDC said. I think it's good to know, right? But we don't.
Yeah,
have
I mean,
the
I outlined
information.
what's probably their rationale, but I
Yeah.
don't agree with it. I would like to see this become a routine part of just wastewater management, where they would just sample and sequence periodic samples of not just for poliovirus. You know, we're now that the pandemic has shown that you can do this, make it part of the standard procedure and keep an eye on what's in there.
So I have a question that would probably be addressed by what Alan just suggested, which is, I mean, we don't know whether this particular outbreak, if you want to call it that, is simply a burst of something that is endemic in the U.S., or whether it was imported from somebody who was vaccinated elsewhere. Do you have any thoughts on that? Just don't know?
We do know that people who've gotten IPV only can transmit it.
Right.
Now, once they do, they've now gotten an oral dose of poliovirus, and they do probably acquire gut immunity at that point. But there are many more people that it can go through. So yeah, it could spread endemically that way. And we also know that people who are immunosuppressed, who get vaccine-derived polio or I guess wild type, if that was around, can continue excreting it for a very, very long time.
But the routine, or at least, more widespread wastewater surveillance could potentially tell us whether this is something that's going on all the time, or whether there are sporadic importations.
Yeah.
Yes.
I think this has been going on since we switched to IPV in 2000, frankly, but we don't know because we haven't looked. But
do you know if there are plans to do this on a larger scale?
Well, my understanding is that the select places, right, these in Philadelphia and Michigan,
right,
and also they will allow states to do it if they're interested on their own, right? Any state could decide to do it.
Sure.
And I would guess the red states won't do it and the blue states will. But
there's also just a funding issue there.
Yeah. So the states will report it to the CDC.
States would have to fund this as part of their normal public health efforts, and they'd have to set up the system for it. And I think there will be some states that are very forward-thinking about public health that will be all over this. And I think they'll be the urbanized, wealthier states.
Yeah.
And I think in more rural areas, probably not going to happen.
So you collect wastewater already, right?
Sure.
For other purposes. So it's a matter of then getting samples and doing PCR. And I know that, for example, in Colorado, Emily Travanti is doing it on her own, right? She wants to know because she's a virologist, right? So, and I think Minnesota, where public health officer is the former eradication chief or something like that, Alan, right?
Yeah, so Minnesota's pretty on top of public health.
Yeah. So, yeah, there'll be certain states that
And
do.
in the New York area, and yeah.
I think it's just very interesting questions. And, some people say, well, polio is under control, but it doesn't matter. You learn things. You can apply them to other viruses as well.
Well, and as I said, it's not just about polio. If you have a system like this and you're doing PCR on sewage, run some more samples. I mean, there's no shortage. It's not like, oh no, I used my last 5 microliters of sewage. You can always get more. So you could sequence for a whole panel of viruses and monitor them all the time.
Yeah. By the way, the group in New York who's doing this, remember Queens College, and we had them on a few times. They're continuing to do this on their own. So that's good. One more thing I wanted to tell you. So a long time ago, maybe a year ago, we talked about efforts to modify OPV2 so it would not revert and become neurovirulent, right? So the isolate from this patient in Rockland County is a recombinant of poliovirus with the 3' half from a Coxsackie virus in the gut. And we know exactly which Coxsackie. And this is what happens with these vaccine OPVs. They recombine in your gut, and within a few weeks, you're shedding actually recombinants, which can acquire neurovirulence just by virtue of the recombination. Anyway, the NOPV2 was engineered to try and reduce reversion. And we also talked of the study of it being in over 100 million kids, and the UTR mutations, the 5' N mutations are not reverting, but it does still recombine with other enteroviruses. So we don't know if it's going to be less transmissible and less neurovirulent yet. It's too soon. Anyway, but what I want to tell you is in the original trial of this, they did a safety trial in the Netherlands in Poliopolis, right? They took some shipping containers and stuck them together and made in the parking lot of a medical center, and they put 15 people in there, and these were people who had been immunized against polio with IPV, okay? Fully immunized, 3 doses. They fed them NOPV2, and they took their stool for the next 100 days, and I left off the time.
As these people were living in shipping containers in the parking lot, yes.
They were. They stayed in there.
And they called the place Poliopolis?
Yeah. They did. They did. In fact, the first paper reporting it was Poliopolis in the title. And so I have a figure here where we look at stool shedding over 100 days, and they do PCR and virus culture in cells, right? So you can see that almost all of these people shed virus for almost 100 days. I mean, it peaks very shortly after they get the NOPV2, and then it goes down, but they're all shedding infectious virus for a long time. And the point I just want to make, these are IPV immunized people. You drink NOPV2, it's reproducing in your gut. So IPV does not immunize the gut. It's very clear. And this had nothing to do with proving that. It was trying to look at the safety of NOPV2. But I remember Amy Rosenfeld pointed this out, and I think it's a good example of that. And in fact, like even OPV, after a few months, you're not going to have much secretory antibody in your gut. And here's one last thing I want to tell you, because yesterday I met with John Udell at NIH. He said the most common immunodeficiency on the planet among humans is IgA immunodeficiency. They don't get any more infections than anybody else. So we're all freaking out over secretory mucosal IgA, right? Now, maybe if it's there, it helps, right? But if it's not there, it doesn't seem to hurt. Okay, so that is what's going on with polio surveillance. I'm sure we'll hear more. All right, now, Rich, I was walking on the NIH campus and I saw the National Library of Medicine, where we went to talk with Eugene Koonin a couple of years ago, remember?
Yep.
And so this is a paper from where Eugene Koonin is the senior corresponding author. it is a cell paper. Mining metatranscriptomes reveals a vast world of viroid-like circular RNAs. The first author is Benjamin Lee. The last author is Eugene Koonin. And this is from the NIH, from the University of Oxford, Tel Aviv University, Lawrence Berkeley National Laboratory, the Pasteur Institute, and Oregon State University.
And one of the authors, there are about a dozen authors listed, but one of them is the RNA Virus Discovery Consortium, which in a footnote you learn is like 50 more people. So this is a big collaboration.
Right. It's a cool paper. So this is all about viroids. Viroids, the smallest and simplest infectious agents. They are covalently closed circular RNA. Right? Circular RNA, small, 220 to 450 bases. They don't encode any proteins. And so they get in, and they don't have a capsid, just naked RNA. And the ones we know about, and after this paper we know about many more, but the ones we know about infect plants, and they can be pathogens in plants, and they go from plant to plant. And it's not something you would say is RNA would do, but because they're so highly base paired, they're rod-like, probably resistant to degradation, or more so
this
than
is
single-strand.
This is the smallest infectious agent known, right? It's the smallest, simplest way that biology has figured out to parasitize.
That's right. He calls it the ultimate parasite.
So they don't code for anything.
Correct.
Right? Aside from the fact that they, I don't know if encode, although they use it, I don't know if encode is the right word here, but they have ribozyme activity.
Some of them have ribozymes, yeah.
All right, because are there examples of these without ribozyme activity?
Yes.
Yeah. Yes, there
are.
So
the basic trick that they pull off is that they have motifs that trick the cell enzymes into copying the RNA as a rolling circle, and then they have sites in them that the cell interprets as targets for cleavage, so that it cleaves off into individual genomes, and the ends of these little 200 base or 250 base genomes are ligase targets, so they get ligated together in circles. So the cell does everything just based on how these things look.
So the real minimalist version of this encodes nothing.
Yes.
Not even a ribozyme activity.
It is just the idea of infection.
Right. But they can nevertheless be pathogenic, right?
Of course, yes, they can.
Because they're a serious distraction.
Yes.
Yes. So some of them encode a ribosome, which, for those of you who don't know, is a sequence of RNA that can cleave itself. You don't need any proteins.
Right. So even though it sounds like an enzyme because it ends in zyme, it's not a protein enzyme.
And then the others that don't have a ribozyme. And you're right, Rich. I don't know if encode is right because there's no protein.
Right.
Yeah. They're not encoding it. They are it.
Right.
Yeah.
Well, we talk about non-coding RNAs. So
right.
And they non-code a ribozyme.
They non-code a ribozyme. Yes.
Well, they say here ribozymes are present in the genome, right?
Yeah.
So that's how they get around
That's a good way of getting around it.
But the ribozymes the viruses that don't have ribozymes, as Alan said, they're cleaved by a host enzyme, RNase 3, Rich. You might remember RNase.
RNase 3 is an old friend of mine.
Yes, I thought so. So these infect plants, and you know what? Some of them can cause disease in plants, and the diseases are named after the plant. We'll get to them, but they're cool names. But the reason they cause disease is because when these viroids get into the plant cell, they're chopped up by the defense mechanism of the plant, the siRNA mechanism, into small siRNAs that target plant cell proteins, and that causes the disease in the plant. So the viroid silences plant genes, and that's what causes the disease. I mean, there's no benefit for the viroid to do that. It's just a byproduct, I guess.
And one of the they're found in plants, and there are only a couple of types of these, but there's one theory has been, well, maybe these are remnants from the RNA world. So the theory of the RNA world, that all life started as RNA that was doing its own enzymatic, its own ribozyme activities, and then it gradually evolved the ability to get proteins into the act and the rest of it. So that's an origin of life theory, which of course is untestable because we can't go back and check, but there's some evidence supporting that. And one idea for viroids is, well, maybe they are remnants from that survived. The flaw in that has long been, well, then why aren't they more common? Why aren't they in more species? Why are they just in a few plant diseases? And that's one of the things this paper is looking at. That do we really know that these are unusual plant-only things?
So these things are transmissible. Is that correct?
Yes.
So like vectored by an insect or something like that.
If a sapsucker picks it up, it could transmit it, but it's also vectored by farm machinery that crushes plants and farmers that walk on them and move them over. Yeah. Well, any mechanical transmission. Yeah.
We made the statement that the siRNA's bit is sort of a sideshow, fortuitous, but I wonder if somehow a sick plant
is more likely to
isn't a better transmitter.
Could be
than a healthy plant.
Because it gets
just wondering.
And there are also ones that don't transmit horizontally like that, the retroviroids, which I had not come across before and kind of blew my mind as I was reading the intro to this, that are reverse transcribed and integrate into the plant genome.
Yeah. Kathy, you were going to say something.
I was just going to say that, Alan described what people have posited, that these things arose from the RNA world because but then there's this thing about, why aren't there more of them? And that this paper is not only going to look into that, it's really going to maybe discount that original theory and hypothesis and allow us to think about other ways that they came around.
Which in Wednesday's lecture, I mentioned that hypothesis, and I'm going to have to change it after reading this paper. But that's science. So, by the way, the viroids, when they get into cells, they're copied by DNA-dependent RNA polymerase, Pol2. Okay. Then there's another group of agents that have covalently closed circular RNA. These include satellite RNAs. Those are copied by RNA-dependent RNA polymerase of a helper virus that needs to infect the same cell. So satellite RNAs, they don't encode protein, but they're packaged by a capsid produced by the helper virus. So they're different from viroids because they actually have a capsid, and they call them encapsidated viroids, which is, I'd never thought of that before, but they don't code for anything. So it makes sense. They have the same kind of structured RNA. They're small. They replicate via rolling circles. So yeah, that makes sense. Encapsulated viroids.
I want to emphasize something you just said in case it slipped by people's radar. You said that some of these are copied by DNA-dependent RNA polymerase. All right? That's an enzyme that's supposed to copy DNA into RNA, but in this case, it's using RNA as a template and making RNA. Go figure. So I don't know how often, aside from this, that DNA-dependent RNA polymerase goes around using RNA as a template, or whether these things have a special trick to make Pol2 do this.
I think these things have a disguise. They go in and they put on their DNA outfits, and the DNA polymerase comes along, and
a lot of them have a lot of structure.
The RNA
structure may be
yes.
part of it. Yeah. But also hepatitis delta is also copied by Pol2. So it's got that rod-like structure. Maybe that's part of it.
Right, and hepatitis delta comes up in this paper as another viroid-like, viroid-adjacent agent, which requires a helper virus.
This is a new word, isn't it? Adjacent, something adjacent.
Yes.
I've just been hearing a lot lately. Just like interrogate a few years ago. I said, what is this interrogate? Now I'm hearing adjacent. What does it mean? Next to?
Yeah, it's always meant to next to, but it is being more widely applied to concepts that would not normally be
Mark Martin,
objects.
who comes on TWiM often, he says, I'm TWiM adjacent. He's not a member, but he's next to it because he's there a lot. That's a funny thing. Okay, you're the second person ever to say that now. But by the way, these satellite RNAs also encode ribozymes. So another feature that makes them similar to viroids. So they're encapsulated viroid. And then there's the retroviroid that Alan mentioned. Oh, here's the one name. Carnation small viroid-like RNA. That's the name of it because it's in carnations, I guess. As Alan said, they don't transmit horizontally, but their RNAs copy the DNA and integrates into the plant genome. So it's transmitted vertically, right? And these are copied to DNA, I presume, by a pararetrovirus, which is a retrovirus of plants that has reverse transcriptase, copies the RNA into DNA. Then we have another group called I thought that the introduction was the best part of this paper.
The
We're
intro
going to
was
spend
quite
most of the time on the introduction.
Yeah.
Retrozymes. I've never heard of this. I don't read enough, I guess. These are now retrotransposons. A retrotransposon is a mobile piece of DNA that moves around the genome via reverse transcriptase. Basically, so the DNA is integrated. It's transcribed to RNA. The RNA is reverse transcribed to DNA, and then it goes to integrate somewhere else in the genome because that's a retrotransposon. And so these retrozymes are circular RNAs that basically move around via reverse transcriptase. They don't encode proteins. They do have ribozymes in them. So where do they get the reverse transcriptase? Well, from another retrotransposon in this cell. Cell's full of retrotransposons. And then, so there's some new classification levels that have come into virology lately, and one of them is a realm. You know, give me a sentence with realm in it from the old days, Alan. The something of the realm, right?
The coin of the realm.
Coin of the realm. Thank you.
Yes.
Perfect. So this realm is ribozyveria. That's the way I think it must be pronounced, right? It's got a Y in it. And I actually should know how high up a realm is. Does anyone know offhand?
No, I'll look it up.
Because we have families, and above families are orders. It must be above orders.
It's got to be above orders, but
This is one of those things that we don't exactly know how to pronounce it, but I was pronouncing it ribozyveria. Ribozyveria.
Ribozyveria.
That's another way I tried it too. It's either ribozy, which makes it sound a little fun, right?
Yeah.
yeah.
Ribozy. And this includes.
Wow, realm is like in the triangle. Realm is at one end. It's realm, kingdom, phylum, class, order, family, genus, species.
So it's above kingdom.
It's above kingdom. It's the biggest of all.
Could it be above kingdom?
I guess. Well,
well, they're domain.
Man, we're going to get mail from Jens. Okay,
Yes.
so
okay.
This to me speaks to this question of whether viruses ought to be their own thing. Okay, so this is almost like saying that viruses are another kingdom.
Yes.
I mean, I'm just looking at the virus taxonomy here. Okay, and this realm, kingdom, phylum, blah, 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 blah. So it could be that in realms, I don't know, there are other kingdoms that are not viral in nature.
Because the kingdoms that we know of, right? We got prokaryotes, we have eukaryotes, and we have archaea, right?
Well, those are domains.
Domains. So what are the kingdoms?
True. When I was taking biology in high school, there was the animals, the plants, the fungi, but
hey, when I took it, there were only animals and plants.
Right. That's all gone out the window.
Oh, you're right. Animals, plants, bacteria.
Kingdoms are underneath domains. You have the three domains, Then you have
Yeah.
kingdoms underneath that, and I'm not sure how many there are at this point.
So you're saying that o-
Realms, though, in
are between-
viruses, I hadn't come across. Now I can't. Nobody can keep up with taxonomy, so I don't know if there are realms in bacteria, in other, like,
I cannot find. I'm looking at a bunch of. If you just Google taxonomy and look at the images, which are all the graphs and stuff, I can't find anything that contains both realm and domain.
all right,
Okay.
yeah.
Okay.
So
write
I
us
don't
a
know what the
letter.
relationship is between
realm
a
may
realm and a
be
domain.
realm may be the viral equivalent of domain. I don't know.
Oh, okay, something like that.
Which would
Right.
imply that the people who came up with this are arguing that viruses are a whole separate thing.
Yeah.
Like you've got you've got your 3 domains of life. We virologists will be over here with our own realms.
Well, it makes
which-
sense if you think that viruses are not alive. You
Yes.
can have your realms of alive things, and then there's this other
Yeah.
realm
Yes.
or whatever.
Jens,
Anyway,
I
we'll
can
hear
hear
from
you
Jens.
screaming.
Yeah,
Yeah.
and write us a email, and we still have one that we didn't read. Actually, we have to
We need to read that.
read that one. So we'll do it anyway. So these riboziviria
Ribosiviria.
includes Delta virus. This hepatitis Delta virus. It's a rod-shaped RNA, highly double-stranded. Encodes one protein though, which is a nucleocapsid protein. It's not a capsid, and it needs the hepatitis B virus glycoprotein in order to package it. So it depends on a helper virus, but it does have a ribozyme. So it's not a viroid. It's different. Ribosiviria, and we did a
Right,
paper.
it's much too ambitious to be
Yes,
a
a
viroid.
viroid.
It actually encodes a protein.
And we used to think that this was the only example of it, but then a couple of years ago they were found in snakes and birds, and we don't even know what the helper virus is. So that's another thing we're going to look at here. Okay, so, and then we get into this theory that Kathy just mentioned, which, they call them minimal replicators or ultimate parasites. And it's been suggested that they're descended from RNA world, but then if that's the case, why are they only in plants, right? So that we're going to look at that. And so that is what they have done here. They have mined RNA transcriptomes.
That's another one of those words that got co-opted. Data mining. How do you mine
I don't
data?
mind that
You-
too much.
with a pick and shovel.
I don't mind. I don't mind it too much.
You don't mine it too much. No.
I mind, for some reason, I minded interrogating, but I guess
Interrogating data. Yeah. Yeah. So this is a bioinformatics paper. They Google taxonomy, and they're going to proceed to analyze an enormous number of sequences from metatranscriptomics, which is just looking at sequencing all of the transcripts in huge
in
samples.
dirt or plants or whatever you got, right? Just
So
Right,
right
it's
off
not
the
like
bat
it's
here,
not like the transcriptome, all the genes transcribed in a human, all the genes transcribed in an E. coli cell. It's all the genes that we can find RNA for in this cup of dirt.
They say they mined metatranscriptomes and plant transcriptomes. Can we please define metatranscriptome?
Right, that's what I was just getting at. So a metatranscriptome is multiple species taken together, and you're looking at the transcripts that they all produce. You could do I assume this is an extension of metagenomics, where you take a fecal sample from a person and you sequence it. Not like, oh, what species are in there? You're doing the sequencing in order to get
it's multiple
taxonomic
species.
groups.
and you got
Or a sample of dirt, or a sample out of
Yes.
Ocean.
Yellowstone,
Sure.
some weird environment.
or the
multiple
nose
species,
of some stray
yeah.
dog you found on the street. Yeah.
So these are going to be mostly prokaryotes.
Yes.
Yeah, but other things are in there, and so that's a metatranscript.
Prokaryotes and fungi.
Prokaryotes and fungi.
And a lot of this is, there's a graph later. Is this open access?
I didn't have any
Oh, it
trouble
is,
getting
yes.
it. Yeah.
Yes.
Yeah, if people want to dig into this, there's a lot of this is dirt samples, but then they've also got some ocean water and other sorts of
Okay.
things.
Some
So-
spruce rhizo something or others.
Right.
Rhizo.
The
what-
spruce
I wrote it down by each of those diagrams.
phylosphere.
Yeah, there it is.
So that
Or a rhizosphere, a phylosphere, and a switchgrass phylosphere, and a spruce rhizosphere.
one of those is from Michigan too, right?
Yes, the
I don't know what
switchgrass phylosphere.
Yeah.
What is a phylosphere? I think I know what a rhizosphere is probably in a rhizome, like a root.
Yeah, it being the root, the phylosphere is the aerial region of the plant colonized by microbes, fungi,
Okay.
algae, protozoa, nematodes, and bacteria, of course.
Right. So it's taking the stems and leaves and just grinding
Yeah.
them up and all the stuff that's on them.
Yeah, and you do transcript sequence, and it's a metatranscriptome because there's multiple species.
It's
right?
multiple species, and you're looking at the RNA in it, the RNA
Yeah.
sequences in it, so it's a transcriptome.
So this is 5,100 metatranscriptomes, billions and billions of bases,
Yes.
and they pulled out 10,000 viroid-like closed circular RNAs, fivefold increase in our knowledge. Okay, so to do this, they use a computational pipeline, and the key to this, I think, is because these genomes are circular, right? So you're going to get concatenamers when you do the sequencing, and so the key part of this pipeline is to figure out how to cleave it, to take the sequence and cut it where you think the circle ends, right? Because it's repeated. So the algorithm obviously looks at the repeats and says, okay, we're going to cut here and do that. And they built this pipeline, and
Which is the same way the viroids replicate.
that's right.
It's kind of they do a rolling circle in order to replicate, and then they get cut into pieces. So they're trying to.
So
Fine,
are
thank
the
you.
sequences that they originally generate, they're not circular, right? They're just fragments, I
Yes.
thought.
Right.
well-
but you know, a lot of these are replicating. The transcripts are concatenamers, right? Because then they're cleaved after they're concatenameric. So I guess they're capturing a lot of those concatenamers.
Okay,
Because
that's
it's
what I wasn't sure they were able to do.
This takes me
Okay.
back to T4 DNA replication. So these must be circularly permuted
Right,
concatenamers.
yes. That's
Okay,
right.
so you could, computationally, you could cleave them anywhere
Yes.
and come up with a circle. Biologically, a ribozyme is probably more site-specific.
It is, yeah. I mean, the ribozyme sequence defines the cleavage site. So you could do that, I suppose. Anyway, so they test it with transcriptomes that contain viroids, and they show that it works. And then they apply it to their metatranscriptomes. 1.5 billion contigs, 708 gigabase pairs. Is that like a hard drive or something?
That's it. That's a lot of sequence.
And so,
So I visualize this procedure as being it's stepwise. They gate on different things. It's kind of like if you do a search and you get too many hits, and then you have the option to set different filters.
yeah.
So they're filtering it down to try to find stuff that's viroid-like.
So the median length is 165 nucleotides. 2.7 million were around the size of viroids, and then 11,378 they classified as viroid-like because they had ribozymes, right? They could predict the ribozyme cleavage site. And then among those, 10,100 had no homology to known viroids, and the rest had some homology with known viroids. So that's cool. It means there's a lot of new stuff in there as well. So then they put all these into clusters, which they have 4,800 clusters, average nucleotide identity clusters, which are all very similar. And then they go into a little depth on 5 of those clusters in this paper. So you could go get the data and look at them yourself. But the 5 largest clusters we have, most of the sequences are similar to each other. Let's go through them quickly. Cluster 1 is about 562 nucleotides long and has a homology to, I just want to say these names, eggplant latent viroid or grapevine hammerhead viroid-like RNA. I just think they're so cool.
And it's not aubergine, it's eggplant.
That's
plant.
right. So, Kathy, if you had discovered that one, you probably called it aubergine, right?
I learned eggplant first.
Okay. Most of these from one came from terrestrial metatranscriptomes from 11 different locations on Earth, 4 freshwater and 1 in a spruce rhizosphere sample. I guess that's the roots, right?
Mm.
And cluster 2, so ribozymes have interesting names, and one of them is hammerhead. And then there's another one here, twister. And there are a couple more names that come up. So this cluster 2 has 68 members. Many of them were found in Michigan, sampling the switchgrass phylosphere. I wonder where that would be, Kathy.
I don't know. I have the materials and methods open, but
Where's switchgrass is everywhere, though, right?
I
Isn't
thought
that
of
a
switchgrass
prairie?
as being in Georgia, but
Okay.
Okay. Yeah, I thought there were switchgrass prairies, and maybe I'm mistaken.
All right. Anyway, cluster 3, 600 bases in length. This one has no detectable match to anything in the viroid database, and most of these come from soil from 6 places in the world: Colombia, Czech Republic, Germany, and the U.S. 2 of them were in water, creek water.
Okay, I've just switchgrass is like this tall grow. There are switchgrasses that people have as ornamental grasses on their landscape. That's where
yeah.
I came across that term.
Okay.
Like everywhere.
Yeah,
Yes.
they're widespread, but I don't know where the natural range of them is. And apparently, somebody sampled a bunch in Michigan.
yeah, and I now see where my confusion is that at least some there's a Spartina that's known as prairie cordgrass, which is different from switchgrass, but there's also Spartina in Georgia.
Okay.
For anybody who cares.
Cluster 5, 450 nucleotides, terrestrial samples, soil, plant litter, and peat, 9 different locations. So those are the 5 largest clusters. So
And these have characteristic secondary structures
that's right.
that are depicted in the figure that talks about the 5 of them. Some of them are like A is they call rod-like, which is like almost completely base paired with no branches at all. It's got looks like it's got a few bubbles in it. Others like B are pretty highly branched, and others are somewhere in between.
Then they found among these hits long ones, long rod-shaped sequences, up to 4,700 nucleotides, which is too long for a viroid or a ribozivirus.
10 times longer than the 200 to 400.
Yeah, so they wanted to know what's going on here. So these had some open reading frames, so they translated them and did some searches and found matches to RNA polymerases, RNA-dependent RNA polymerases of single-stranded RNA viruses called ambiviruses that were recently discovered in fungi. 4 kb genomes, they have open reading frames, one of which encodes an RNA polymerase. And so they think that these are actually ambiviruses. This is a distinct ribozi-like viruses that have a RNA-dependent RNA polymerase, which previously had been discovered as riboziviruses, but they are circular and have ribozymes. So that's something new.
If I were designing all this, I would make the other open reading frame in those guys some sort of structural protein, but they don't comment on that.
You would make it that way. Well, you're thinking about it. Evolution doesn't think; it just reacts, right? Then they find some homology with mitovirus, RNA-dependent RNA polymerase. Mitoviruses reproduce in mitochondria. They're RNA viruses, and so they're finding some hits with those. And they also find hits with this is with homology to capsid proteins of single-stranded DNA viruses. These are highly abundant single-stranded DNA viruses. It's called CRESS viruses. What does CRESS stand for again? I can't remember. Should we look it up?
Is it just lowercase CRESS?
Yeah.
Well, that's a kind of
That's
plant.
Yeah, it's a plant.
No,
Like
not the plant.
watercress.
It stands for some
Oh.
single standard.
Oh, okay. That's why I asked if it was
Here.
lowercase.
Eukaryotic circular rep encoding single-stranded DNA, crest DNA.
Okay.
So rep is the protein that gets the polymerase to be attracted to the genome. So they find that these look like crest viruses, and they have ribozymes as well. So this is totally new stuff. A distinct group of ribosy-like satellite viruses. And they look for delta. They find a lot of delta-like viruses. Remember, hepatitis delta depends on hepatitis B virus as a helper. They find a whole bunch of these in soil, wastewater, coastal wetland from Alaska to Florida. They have ribozymes. They have rod shapes. They encode proteins roughly the size of hepatitis delta virus. And then they find, so most of the viroids we know about so far have one ribozyme. And then they find examples where there's two and multiple, in two different kinds, different combinations. So see, almost all viroid-like RNAs described to date contain the same type of ribozyme on both polarities, of course. And they found ones where it had different kinds of ribozymes. And they found twister ribozymes. It's a certain sequence motif. They called it twister. I don't know why they called it twister. Another one called hatchet. Another one called pistol.
I think these are probably named after the shapes
Maybe.
of the
Because
2D
hammerhead
structure.
certainly is,
Yeah,
right?
hammerhead is, and I'm assuming the rest of these have some shape.
So
that
now,
is
hammerhead,
suggestive.
twister, pistol, and hatchet. Kind of like that they're creative, right?
Yes.
Yeah.
It's like the Drosophila geneticists,
Yeah.
right? Who
Right.
name genes cool ways. And then the other thing they do is ask, okay, so the CRISPR defense machinery in prokaryotes works by when the bacteria is invaded by a DNA or a phage, it takes a piece of that and stores it in the genome, and then that's made into RNA later, which will be used to block the incoming phage or DNA. So if you sequence the genome, you might get an idea of what infected it over time. So you could theoretically match a viroid with its host.
So essentially, they're doing a serosurvey on bacteria.
Yeah, but not for antibodies, though.
No, not for antibodies. They're looking for what the immune system of the bacteria, which
Right.
we now know exists, might have responded to in the past.
So all these sequences they have now, they can search against the CRISPR database, and they find hits. They found 89 spacers with matches. So 89 sequences in bacteria that match these viroid-like RNAs. And then now you see, viroids can infect bacteria. Wow, we didn't know that. Well, I didn't know that anyway. Maybe someone knew it.
That's a
on
whole
Earth.
other domain.
Whole other domain. And so they have interesting pairs where they find a viroid in a hot spring, and then the CRISPR is 800 kilometers away, which is just, that's the way it is. It didn't move, but you just don't have that much sampling, I guess. So they find some matches suggesting that some of these viroids infect specific hosts. I mean, they have matches from, for example, a sludge bioreactor. So somewhere in that bioreactor is the host for this viroid. You don't know which one it is, but because these are metagenomes now. They're just mushes of the whole sludge reactor, right? You take sludge and you sequence it. And then where are these located? Throughout the world, all types of ecosystems, mostly soil samples, but they get them from other, as we said, plants and waters as well.
My guess is anywhere you look.
Anywhere you
Yeah.
look, you're going to find them.
Yeah, because the sampling map is kind of a map of where researchers work. A lot of samples in the U.S. and Europe, and then it's kind of scattered in other places around the world, and some in oceans, but nowhere near representative sampling from that. And given the diversity of where they're finding these, I would agree with Rich. I think they're going to be everywhere.
And so they say most of these seem to be infecting prokaryotes. they say it's really hard to know, but they say most of the transcriptomes that they looked at had less than 0.1% eukaryotic sequences or contigs. And they say they're either very rare or undetected in eukaryotes, mostly in prokaryotes. And they said this is not known, that was not known before. And also they're everywhere. They basically, the host range, the ecological range is much bigger than we've appreciated before. Okay, so that's it. I think you discover a lot of new viroids. You find that ambiviruses are actually viroid-like agents. Viroid-like mitoviruses and satellite viruses, a lot of prokaryotes. Bacteria support a much greater diversity of RNA replicators than previously suspected. So, it's interesting. Just the other day, I gave a lecture in my course, and DNA viruses dominate the prokaryotic virome, but RNA viruses do not. But now we're seeing from this a lot of viroids in bacteria, which is really interesting. Why not RNA viruses? There's a theory for that, but I don't remember what it is.
So how does this inform one of our initial questions about the RNA world?
So they're not just in plants. Viroids are not just in plants. And so they are everywhere, as you might expect. If they were present in an RNA world, which was ancestral to everything, they should be in many different species. And in fact, that's what we see here.
Yeah.
But they do say, and let me find that paragraph. I read it 3 or 4 times and I had trouble understanding it, but maybe we can crowd understand it. Here we go. That happens on TWiV a lot, right?
Mm-hmm.
Well, I have to say, I was going to comment that I read this paper and thought, this is typically crazy, all right. But discussing it with you guys, I'm sitting here getting all pumped up and think this is really crazy.
Yes.
This is crazier and crazier.
I have to find it now. Where is it? Is it in the introduction?
I looked for that paragraph earlier and could not find it. If we're talking about the same one,
Probably.
I thought I even annotated on paper in the margin, but I can't find it.
Here it is. It's in the introduction. So, given the apparent narrow range of host range of viroids in plants is poorly compatible with this evolutionary scenario from the RNA world. And then they say, instead, given the similarities between retrozymes and avsun viroids, it has been suggested that avsun viroids descended from retrozymes. And I'm like, huh? What? So a retrozyme are CCC RNAs that propagate by retrotransposons, right? They don't encode proteins, but they hijack the replication machinery of other retrotransposons. So avsun viroids, which is a kind of viroid, is descended from. So what they're saying is retrozymes are more ancient. So we didn't go from RNA world to plant viroids, in other words. So there's an intermediate and retro.
Right, went RNA world to other more complex life, and then these retrotransposon elements evolved viroids. Is the idea.
Yeah.
So I may have misunderstood it because I couldn't go back and reread I didn't go back and reread the introduction, but they were saying that there was this idea that they're direct descendants from primordial RNA replicators, but the problem was that they had only been reported in plants.
Mm-hmm.
Right.
But
Right.
now, if they've been reported more widely, maybe we can go back to the direct descendants from primordial RNA replicators. Do I have that right?
But they also argue in the discussion. Oh no, that's different. Never mind. That's about the hepatitis delta and relatives. Right, the ribosi viruses.
In a way, I don't care if they're direct descendants or whatever,
Yeah,
because
I
there's
know.
just this complete sea of these things out there.
Yes.
Okay. I mean, the thing that overwhelms me with this is that we got little pieces of RNA that don't code for anything, copying themselves all over the place.
All over
Right.
the place.
Now,
And
Yeah.
Granted, they have assistance from an evolved organism, but
so the crux of the matter is, did this evolve independently in these different places, and it's just a very commonly evolved strategy, or is this something that existed in the RNA world that continued to persist and branched along with all these forms of life?
right. And I don't think this necessarily gives us any information about that, but I have a hard time believing that hasn't been going on since the beginning.
And also, it might be hard to distinguish between those two if this is something that can very commonly evolve, which I would think when you're dealing with 250, 300 base pieces, it's not like you got to change a lot to get a whole selection of all of your possible sequences, right?
Yeah, there's no actual phylogeny on these things, right?
Right.
These are just, at this point at least, and it may be hard with sequences that are so small.
I
even
think
to
you may
create
not
such
be
a
able
thing.
to, yeah, how do you construct a tree when there are so few bases?
Crazy. It's just crazy. The whole thing is just nuts.
But I did find an image and pasted in here for you guys of a
Yes.
pistol ribozyme.
Pistol ribozyme.
But I couldn't find any image of a hatchet ribozyme that looks like a hatchet.
Pistol ribozyme, the way it's colored here, looks
Oh.
like something out of a sci-fi cartoon.
That's very
It's
cool.
Definitely.
Well, it also
pistol
looks
shaped.
kind of like the tRNA 3D structure. Just
It does.
Okay, sure.
I'm reading a paper here on retrozymes while you guys are talking.
Maybe we should get back to doing TWiV.
These are propagated through retrotransposition. Very interesting. I have to read more about that. All right, back to TWiV. All right, we're done with that paper, but that's quite and that's cool. The pistol is neat. It's beautiful. Nice picture. All right, let's do some email here.
I have to tell you,
Yeah.
because I've been thinking about this. We've been talking about it. Harper, remember Harper,
Yeah, I do.
who is now 14
Whoa.
and in
Wow.
high school. She's learning about the genetic code. And I'm
Great.
just so pumped.
Yeah.
She starts talking about, I think, in fact, she was, according to her syllabus, I think she was going to be learning about viruses today. Right?
Nice.
And so I was having this conversation with her in the car as we were driving to dance, as we have done forever. And I said, the cool thing is that the code is the same in virtually in every living thing on the planet. What does that tell you? And then we came home afterwards. We're sitting around with me and Harper and my wife, and this subject comes up again of her studying the code. And my wife says, the cool thing is that the code is the same in everything. So that got drilled home.
Yep.
It's just awesome.
good.
This is just so crazy.
All right, Alan, can you take the first email, please? I shortened it up a
Okay.
bit.
Oh, yeah, I see. John writes, good evening to all the TWiV crew from the land of enchantment, New Mexico. It's a fairly warm 60 degrees Fahrenheit, 16°C out here this afternoon. The weather has generally been freezing at night and warming to the mid-40s to low 50s in the afternoon. We had a lot of rain last weekend, and it was actually foggy this morning. I had to be a bit careful on my morning 3-mile jog because there's still some moisture in places in the street, and some places on the sidewalk felt a teeny bit slippery. I just finished listening to TWiV 958 and thought I would offer my input on the spent nuclear fuel and other waste problem. This is, by the way, This Week in Virology, but yes, we do this. For background, I served 20 years in the Air Force working on a bit of everything from a missile launch officer to managing recording equipment for some of the last underground nuclear effects tests to being responsible for safety and security on our current nuclear weapons. I have a BS in physics from Michigan State, an MS in nuclear effects from the Air Force's own graduate school, AFIT, and I did 3 years of graduate work for a PhD in nuclear engineering at the University of Michigan, where I passed my candidacy exam but didn't have time to do the actual research. Hail to the victors and good luck in the playoffs, Kathy. I haven't done any actual nuclear work for a long time, but thought I could offer some additional sources and input for you. For starters, U.S. policy is governed by 42 U.S.C. 10222 Nuclear Waste Fund, which established a fund based on fees charged to nuclear power generators starting back in the 80s. The fund is currently valued at about $44 billion. The original plan was to select 2 sites for long-term storage of spent nuclear fuel, one east of the Mississippi and one to the west. No suitable location could be found east of the Mississippi, and that was scuttled. Yucca Mountain, which is located on the Nevada test site, was chosen for the western location. A lot of research was done on how to store waste there. However, that was blocked mainly by Nevada's senators, and further work has been halted. There is also objection from Native American tribes in the area. There is also the Waste Isolation Power Plan, WIP, located near Carlsbad in southeastern New Mexico. This is dedicated to storing defense-related waste from various locations, including Los Alamos and Livermore Laboratories, and he gives a link. A good starting place for learning about the topic is the Wikipedia article on radioactive waste, gives a link to that. Several countries are currently reprocessing spent fuel and using it in reactors. In the United States, this used fuel is usually stored, while in other countries such as Russia, the United Kingdom, France, Japan, and India, the fuel is reprocessed to remove the fission products, and the fuel can then be reused. The fission products removed from the fuel are a concentrated form of high-level waste, as are the chemicals used in the process. While most countries reprocess the fuel carrying out single plutonium cycles, India is planning multiple plutonium recycling schemes, and Russia pursues closed cycle. I think you mentioned a link to Orano, which is the French-based international company that manages waste recycling and disposal. For what it's worth, La Hague is located at the far northwest corner of France, and gives a link. They have some operations in the U.S., which seem to involve extracting or otherwise producing radioisotopes for medical and other uses. Other countries are also disposing of nuclear waste. As I noted, the U.S. has no active plan for disposal of high-level waste, but several other countries are developing storage sites for that. And then we're going to skip a large section. Special thanks to Dixon for his jazz project. I certainly agree with his choices. 2 of my personal favorites are Nina Simone and Sarah Vaughan. I especially like Sarah's live concert in Tokyo. I'll have to start listening to some Betty Carter. One of the highlights of my time at U of M was attending a live concert by Linda Ronstadt with the Nelson Riddle Orchestra. I also got to enjoy a performance by jazz flutist Paul Horn at a small club here in Albuquerque a few years later. Wishing you all a good week, John. Thank you, John.
So the U.S. has no active plan for disposal of high-level waste.
So we just are parking it in so-called temporary facilities until somebody figures out what to do with it.
Oh, Congress is going to figure that out.
Oh, yeah. Yeah, they're on it. Absolutely. Our highly functional legislative body.
As soon as they figure out immigration.
Yes.
Anyway, there's more to this letter if you want to read it, a lot more, but it was just
That'll
too
be
long
online.
to read. Thank you, John, for that. Rich.
Craig writes, something I wanted to add to the APOD Artemis photo. Is this the one where we had the moon in the foreground and the Earth in the background, or something like that?
I'll look it up.
We had a couple. Note how dark the moon is. That's the true color. Contrast with the white of the spacecraft and the white of the Earth clouds. Yeah, I think this is the one where it's a sort of a selfie from the spacecraft. You see a bit of the spacecraft and then the moon and the Earth behind it. Mm-hmm. It looks bright white in our night sky because we don't have something whiter to compare it to. I was interested to hear about John Campbell. I had subscribed and respected and learned from his video early in the pandemic, but around when vaccines became available, I started wondering about what he was saying and started skipping the videos. Sometime after that, I thought his titles were odd and continued to not watch. Now I see it wasn't just me, and I'll go ahead and unsubscribe him and subscribe to Debunk the Funk. So that must have been the episode where we had, what was his name, Wilson?
Dan Wilson.
Dan Wilson on Debunk the Funk. That was great. The discussion of the word funk and mention of Australia, I listened to Linguistics podcast even though they harm me, having killed many of my pet peeves. One is from Australia, gives a link, which was preceded by another podcast. He's got a couple of linguistic podcast links. Boy, he's got time on his hands. A related tangent, anyone who lives in Massachusetts can get a Boston Public Library card, which, among other things, gives you online access to the Oxford English Dictionary. That had been one of the things I most missed leaving academia, the OED access. So I was thrilled to find this. While I'm here, back on TWiV 931, you mentioned Keith Jarrett and the Curl concert. You may be interested in this episode of another excellent podcast, Cautionary Tales, which gives the backstory of that concert, organized by a girl 17 years old at the time, in the context of how creativity can be fueled by constraints and challenges, and gives a link to that. Finally, I love the intro, the kind that make you sick, and the outro, another TWiV is viral. They're so lame, they're exactly what I would come up with. You mentioned your drop in Patreon. I'm not in great shape, but good enough that that's inspired me to look at starting to contribute. Craig. Oh, here he's got a link to the APOD picture. So,
I put that in. Yeah.
Oh, you
Kathy
put that
put it
in. Yes.
Mm-hmm.
Okay.
I don't.
So thank you.
I don't
Craig.
think they're so lame. Do you guys think they're lame?
No.
No, I
they're
think
good
he
puns.
means in like a dad joke kind of way.
Yeah.
Oh,
That intro and outro have survived since day one. Is that right?
Yeah.
Yeah,
I think they're cool. I just
Yeah.
like them.
Yeah.
we're not going to change him now.
I mean, I just was editing the first episode and I came to mind.
Mm-hmm.
There you go.
Sometimes the best things come in a crunch, right?
Mm-hmm.
All right, Kathy, please.
Rachel writes, Hi TWiV team. I'm about to graduate med school and I've been listening to TWiV since the beginning of the pandemic because of my interest in infectious disease. About a year ago, I introduced my grandpa, a retired pediatrician, to the podcast, and he's been listening religiously since. He and my grandma have managed to avoid COVID until this week when he got sick, and she followed a few days later. Both are as vaccinated as possible. He qualifies for Paxlovid and is finishing up his course, but she does not due to some comorbid heart conditions. Anyway, long story short is that her doctor was going to give her a monoclonal antibody infusion, but because my grandpa is up to date on TWiV, he knew it was no longer recommended. The doctor asked around and concurred, so now my grandma is being given an antiviral pill she doesn't remember the name of, but I suspect it's molnupiravir. Her geriatrician will be checking in on her daily. Just wanted to pop in and say, keep up the good work, Rachel.
Outstanding.
That's a nice
Excellent.
story. Yeah.
I mean, not every doctor will listen and go,
Yeah.
look, right?
Right.
Right. Good for him.
Good. Well, I think you should be open-minded as a physician, right? You shouldn't take the attitude, well, you found this on the internet, so I'm not going to think about it. Why not, right?
I think most physicians are probably open-minded to reasonable stuff that patients might bring up, especially if the patient happens to be a retired physician.
Yeah, I mean, I also think that the new generation of physicians is coming up with the internet, so they're more likely to accept it too. John writes, I wasn't expecting to write anything about epitope 973 until NYC Paul's letter came up about the mechanical aspects of biology with reference to how Spike manages to dock with ACE2. I think he's probably starting from the lock and key conceptualization of how enzymes work. Not only do substrates manage to find the catalytic sites of their enzymes, but the speed with which some achieve this is staggering. Carbonic anhydrase was the old standard champion reference with a maximal turnover of around 600,000 molecules of carbon dioxide converted to bicarbonate per second. But catalase has beaten that. There are more interesting examples in the paper below, like the collective length of DNA being synthesized per 1.3 seconds by the entire human population relative to the orbit of Pluto. Have a look.
Wow.
I love the turnover numbers. Isn't that incredible per second? Enzymologists have struggled to explain how enzymes with even more sedate turnover rates manage to bind their substrates with such fidelity and speed? I suppose it all gets down to the electronic character of the substrate and binding site. Ionic interactions, van der Waals forces, and the hydrophobic effect surely play major roles besides just the shape of the pocket. Daniel Koshland gave that the cover term orbital steering. Wasn't that a feature of the 58 Oldsmobile? And someone else came up with the term propinquity. And then there's induced fit. I remember that one.
Yeah.
But these were all things that were mentioned in the first lecture on enzymology, after which we moved on, which makes me think that it was all largely formalized hand waving. That was all before molecular modeling, so maybe more insights have emerged. In any event, knowing the binding rate and equilibrium constant between the SARS spike and ACE2 could perhaps give some context to terms like infectivity and transmissibility, but I doubt anyone has spent any time on this. Well, funny you should say that. It could, but no one, I mean, people just assume higher affinity means more infectivity and transmissibility. But I think you need to prove it. You need to do some experiments. And
I could
higher
argue
affinity.
the opposite.
Yeah, exactly. Higher affinity, the
Yeah.
virus could get stuck there
Yeah.
and not be able to move, and then that's a problem.
But you know, even if you did it in cell culture, you just don't know if it would
You
be
still
the
don't know.
same
Yeah.
No.
people. But you know, you can't guess just because you can't do the, well, you could guess, but you can't say this is right. Right?
You have to say it's a guess.
Yeah. All right. So Koshland, by the way, he's got a little asterisk on Daniel Koshland, by the way, was great. He was a protein chemist. Back around 2000, he was scheduled to give the keynote address at the annual Protein Society meeting, but he didn't show up. Nobody could reach him. He was around 80 at the time, so everyone was worried. He showed up the next day, having gotten his days mixed up. In starting his talk, he apologized profusely and said that to atone, he thought it would only be fitting that he should be lowered into a vat of boiling oil and his remains buried in the field with paupers and nucleic acid chemists. This is just, you can't make this stuff up. Further, just now checking his Wikipedia page to refresh my memory of some details, I was astonished to learn a few more things, like that in 97 he was listed as the 64th wealthiest person in the U.S., derived from his father's role in rescuing Levi Strauss in the Depression, and that he had been with the Manhattan Project.
Wow.
Wow, I didn't know that. Oh, otherwise, all in all, a better than expected, nice and sunny afternoon that got up to 5C in Greater Braddock, PA, exclamation, because we didn't know where it was, where tomorrow's sunrise, like yours, will be the latest of the year, with sunset now coming 15 minutes later than its earliest back on December 13. It was a good afternoon to be up in a tree with my new 40-volt battery-powered Cobalt chainsaw from Lowe's. No more fooling with the carburetor, getting it warmed up and then getting it running again while up in a tree. Best regards, John. You know, I hate chainsaws. They're so hard to start. I haven't had one in many years because I could never get them started.
I have an electric chainsaw. It's a plug-in electric chainsaw. When we bought this house, it quickly became apparent that some of the yard work was going to require something more than just a hatchet, and and I deliberately got an electric because I thought, gosh, I'm going to use this thing like once every 3 years. There is no way that a gasoline engine is going to be functional when I need it to be, and I'm so glad I got an electric. I can go out. It's been sitting in the shed for ages, and I know I can go out there, plug the thing in, and just whiz it up.
I don't need any more chainsaws. I hire people now. That's it. I'm done.
Mostly I hire people, but back a few years ago when we had an ice storm in October, when all the leaves were still on the trees, I called it the tree apocalypse because we were offline
Remember.
and had no power for 8 days.
Yeah.
The street was completely blocked with trees. Nobody could go anywhere, and I had to cut my way out of the driveway with my electric chainsaw, which I was able to run from a generator, which I also have.
That was the year of Sandy because that happened 12 days after Sandy.
Oh, we had, actually, it was 2011 that we had our
It wasn't. Sandy was 2012, right?
Yeah.
So
Yeah, by that time, we had
okay,
all the stuff that was going to fall in our neighborhood had already fallen.
because we had Sandy, and then 12 days later it snowed.
Yeah.
It was
Right.
early in October. It was very strange. Anyway, this is a letter full of just interesting stuff.
Very cool.
Yeah, yeah.
Love it.
Being buried with the paupers and the nucleic
Yes.
acid.
It's like as in chemists.
Yeah. All right. Thank you. That's cool. And let's do some picks now. We have so many good emails. Maybe we should do an email episode. We haven't done that in a while.
Not for
Yeah.
a long, long time.
Oh, yeah. Pre-pandemic, probably.
Yeah, I think so. Well, no, we
Yes.
did some
Did we?
email.
Oh, yeah, because we had so
pandemic
much
episodes
pandemic email.
where
Yeah.
we were just answering pandemic related emails. But
Yeah.
good old fashioned TWiV email episodes.
I just got
good.
the sense that people didn't like them as much as regular episodes, but I like them because in each one can be embedded entire discussions.
Yeah.
Right?
There could
Yeah.
some of our best and most interesting discussions came up from emails from people,
Yeah.
usually starting off with the phrase, "This is probably a stupid question, but," and then 45 minutes
Yeah.
later, we're still discussing their excellent question.
All right. Well, maybe we'll do one soon. Okay, let's do some picks. Kathy, what do you have for us?
I have two astronomical picks. One is a blurb about a new dark energy camera in Chile that captured an image of the Milky Way that reveals 3 billion objects that were previously uncharted. So 3 billion new things that they couldn't see before because of this camera, the dark energy camera.
Hey, it's like
just got a
viroids, huh?
picture of it. Yeah.
Yeah.
Yeah, kind of. And then the second one is today's APOD. Brianne picked the How to Find the Green Comet article, and this is a nice picture of the green comet. Someone asked me recently, have there been any pictures of it? And I said, I haven't seen any yet, but here's this one.
Nice.
Pretty nice.
It's very nice. Yep.
Yeah.
And lots of points in the background. Look at that.
Hmm.
I wonder how many are in that one field. Thousands, right?
Yeah.
So cool. Rich, what do you have for us?
If I could spend a moment on
Yeah.
Nick.
Yep.
So this is an obituary of a longtime colleague of mine and a friend and or acquaintance of others here on the show, Nick Musichka, who died on January 6th after a long battle with cancer. He was 75 years old. So Nick was spent part of his career at the University of Florida, so I overlapped with him. I never directly collaborated with him, but I served on committees for some of his students and et cetera. He was a very close collaborator of, oh man, blanking on her name. Come on, who died recently of
Oh, Mavis.
Mavis.
Sorry, very close collaborator of Mavis, and close collaborator of sort of career collaborator with Ken Burns.
Mhm.
He came down to UF essentially with Ken Burns when Ken Burns took the chairmanship. I just want to read some of this stuff because this is an obituary in the local paper, the Gainesville Sun, because it's fascinating. He was born in Germany, son of Ukrainian parents who were brought to Germany during World War II as forced labor. After the war, they lived in refugee camps until they immigrated to the U.S. when Nick was 3 years old. So Nick spent his first 3 years in a refugee camp in Germany and is of Ukrainian descent. Family lived in New Jersey. He got his PhD at Johns Hopkins and did a postdoc at Johns Hopkins. And in 1980, his lab started the development of adeno-associated virus as gene therapy vectors, and explained his life's work as a quest to find a way to help those with genetic diseases using AAV virus as a gene delivery system. So it's fair to say that Nick is a prime mover in the use of AAV as a gene therapy vector, and it is one of the most prominent gene therapy vectors out there. I should also add, it's not here in this obituary, that his basic work on replication of AAV is some of the best hard basic science I've ever seen. It's really great stuff. So it goes on to summarize some of his accomplishments at UF. He founded the Powell Gene Therapy Center at the University of Florida. He also started a company called Applied Genetic Technologies Corporation that does gene therapy stuff. In the last paragraph, I love this.
Mm-hmm.
In accordance with his wishes, no memorial service is planned. However, we, his family, ask that in Nick's memory, remember the many who have and continue to flee war-torn, impoverished, and politically oppressive areas of the world. Remember that immigrants from all over the world of all races, religions, genders, and ethnicities should be welcomed and given a chance to live and thrive free from fear. So I can't say any more than that. Nick was a great guy, and we'll miss him.
Yeah.
Yeah, it's too bad. I knew him also. We all did, and it's
Mm-hmm.
really too bad. I didn't know he was sick.
My pick is an article that was published in, hang on, I've got a letter here. I had some correspondence with Felicia Goodrum, the first author on this. Came out yesterday.
Mhm.
It's in JVI, MBio, and MSphere. Felicia's the lead author because she and Jim Allwine conceived it and wrote the first drafts and recruited help of others. There are 6 authors before the list goes alphabetical, who are Anna Sloan, Seema Lakdavala, Jim Allwine, Arturo Casadevall, Mike Imperiali. And there are a total of 156 authors on this. And it is titled Virology Under the Microscope, A Call for Rational Discourse. I recommend it for people reading it. And it basically, actually, I highlighted some stuff on a PDF here. Excuse me, let me get it up.
It's open access.
Yeah, it is open access. Importantly, they say a small but vocal group of individuals has, so it talks about how science really helped bring or keep the pandemic under control. And that the pandemic has brought a lot of attention to virology. And the attention is focused on the concern about the safe conduct of research with human pathogens. A small but vocal group of individuals has seized upon these concerns, conflating legitimate questions about safely conducting virus-related research with uncertainties over the origins of SARS-CoV-2. The result has fueled public confusion and, in many cases, ill-informed condemnation of virology. With this article, we seek to promote a return to rational discourse. And they proceed to basically go over arguments about some of the key issues, origins of SARS-CoV-2, gain-of-function research, and they talk about how scientists perceive these things and what safety measures are in place to address some of the concerns that have been voiced. An important sentence here towards the end is, given the scientific complexities involved, decisions surrounding these things should not be legislated. They should instead be made after full consideration by scientists with the relevant expertise, with appropriate oversight from Congress. It outlines the problems quite well and the measures that are in place to deal with some of the problems, and I think does what they want to do, which is create a focus for rational discourse going down the road. So I commend this to your attention.
Great. And when we had Felicia on the show a few weeks ago, she I think, plugged this, right,
Yeah.
Vincent?
Yes, she
Yeah.
did. Yep. Said it was in
So
press. Right?
it wasn't out at that point, but she said she was working on this, and yeah,
it's
so
good.
excellent pick.
Rational discourse.
We'll see.
We'd like to see some of that in many spheres, and yeah,
We will see.
I mean, that's what we need, but unfortunately, many people are not rational.
Well, we can help
Yes. we'll
in
try.
whatever way we can.
Can you tell Marjorie Taylor that it's not a so-called pandemic?
Sure. She's
good luck
she's
with that.
right here in my Rolodex.
right.
Oh my gosh. All right, my pick. No, I'm sorry. Alan is next. I'm very sorry, Alan. What do you
That's all
have
right.
for
No,
us?
It's quite all right. Oh, by the way, I wanted to mention up near the top of the show, you asked how many people are on our Discord server, and it was 307 at that time. It is now 309.
Cool.
So
Excellent. Very good.
yeah. And yeah, so my pick, I've been picking indie story-driven video games that people might enjoy, and this is my series. Since people were doing series, this is the third of them. And by the way, I'm providing links to the Steam game store for these, but these are actually available on all platforms. In fact, I think I played this one on the Nintendo Switch. This is Valiant Hearts: The Great War. It is unlike any war-related game you've probably ever played. You know, it's not some first-person shooter where you're trying to kill a bunch of people. It's more like a third-person helper. You're trying to guide these characters through this horrifying situation of World War One. And it is obviously it's fictionalized, and in order to gamify some elements of it, they took quite a few liberties, but it is based on real events. And in fact, there are options in the game where you can look at little historical footnotes to find out about trench warfare or the Gurkha troops, or, you know, various things that come up in the course of it. It is a beautifully illustrated game. The artwork is amazing. It's this watercolor type of palette, and very simply told, but extremely emotionally intense story. The opening sequence of it almost had me in tears, and the closing sequence of it, somebody was cutting up an onion in the next room. I think something was definitely wrong with my eyes. Really, really neat game, and I highly recommend it. Even if you're not into video games, this is not, you know, like Twitchy Reflex Challenge. This is a story told in the form of a video game that makes you a participant, and it is very well done.
Cool. Twitchy reflex.
Like
yeah, that's the stuff we don't have anymore, right?
That's right. No, I totally appreciate that. That's one of the things that draws me to these games that have really good stories behind them. I mean, also I'm a writer, but this is a great example of games as art, I think.
So, Alan, I thought you were going to give us the statistics about the rate of DNA synthesis and the length and so forth. So I looked it up just now. The
right.
rate of DNA synthesis, if extrapolated for the entire human population in terms of total DNA length, would be able to trace the orbit of Pluto in just 1.3 seconds.
That was from the letter, right?
Yes, that was from John's letter, the same letter that had the tidbits about Koshland. Yeah.
I get to look that up.
Yeah.
Neat.
Yeah.
My pick is the Vaccines and Related Biological Products Advisory Committee meeting of January 26th. That's yesterday. I have a link to the PDF, which has all the materials that were presented at the meeting. And this is all about future vaccination regimens for COVID-19. So a lot of material here. And basically, there was one voting question put to the committee, which was, does the committee recommend harmonizing the vaccine strain composition of primary series and booster doses in the U.S. to a single composition? The composition for all vaccines administered currently would be a bivalent vaccine, original plus Omicron BA.4/5. 21 out of 21 said sure, and pull off it there. So he voted yes.
So wait a minute, what would the composition be?
Well, it would be a bivalent vaccine. for
Still
everything.
containing some ancestral, or
Yes.
Yeah,
Right.
ancestral
Original-
plus Omicron. But you remember, right now we have 3 ancestral, 2
Yeah,
ancestral, and the
right.
booster would be bivalent. Now it's
Right.
all bivalent, right?
Right.
For everybody. So people are getting vaccine for the first time, they'd get bivalent. Yeah.
So well, so the thinking is we know that the ancestral confers immunity, and we don't want to let go of that. We aren't going to go the whole way. We aren't going to go totally whatever the currently circulating variant.
Well, that's but then the third bullet point includes, do we need to have the original in there?
Yeah. Right.
So they wanted some discussion on the vaccine composition. So the other 2 bullet points here are discussion points. They didn't have a voting, but one of them was future vaccination campaigns. Should we simplify the schedule for 2 doses in young children and older adults, or 1 dose in all others? So questions about that. And then the composition, discuss and provide input on the consideration of periodic updates. And one of the issues was, do we need the original in there?
Right.
And, at the moment we don't have data, and some of the discussion was around, we need more data, and please don't just give us neutralizing data anymore. We need T-cell data. We need FC effector function data as well. Please, not just neutralization, which is really good.
Good.
So that's the just a little summary, and the document has all the background, and you can probably go and get the video as well. Florencia, they have a listener pick from Florencia who writes, I enjoyed the pick series you're doing about authors who write detective novels. My husband and I are fans of Wallander. And I also remember reading Red Harvest too many years ago. I am very delighted that you picked Andrea Camilleri with his Commissaro Montalbano. This character is an absolute delight, and all that surrounds these novels too, an extremely enjoyable reading. I frankly started typing this email to send Camilleri as a pick since I thought you would enjoy it, and I had not heard it come up yet. I'm about 1 month backlogged on my TWiV listening, so this explains it all. Then I had the brilliant idea to go check on your recent picks and was quite delighted to see it there. Since I am writing, I just read this story on NPR relating Hendra virus, which is very tied up with bat ecology and habitat preservation, which has been a source of so many recent interesting discussions and guests on TWiV. So Florencia provides a link to that NPR article on goats and soda, and also the Nature article. Now that I broke the ice and wrote for the first time about 2 weeks ago, you may never get rid of me. Ha ha. Thanks for your amazing work. Florencia is a professor at Mississippi State University. And that will do it for TWiV 979. You can find the show notes at microbe.tv/TWiV. You can send us a question or a comment. We'd love to get it, or a pick of the week, TWiV at microbe.tv. If you enjoy our work, we'd love to have your financial support. Microbe.tv/TWiV. Kathy Spindlers is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.